Um, Welcome everyone once again to the Greener Hub webinar series. My name is Evelyn Zuniga and I am one of the communications officers for the Greener Hub project. If you have been here before, you know that the Greener Hub project officially started in 2022 as a coordination and support action of the European Commission under the Horizon Europe program, and it currently represents 15 ERANETs, co-funds, and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs last year in October, and the first topic of our series was sustainability in agriculture. Then we moved to another topic, which was biotechnology to advance sustainable solutions for bioeconomy. So if you're interested in any of these topics like mixed farming system, mitigation of greenhouse gas emissions, sustainable farming, production of bioplastic, production of green chemicals, among others, all of our recordings are now available in YouTube. And now in this series, we are talking about sustainable food systems, and we have been talking about food resilience and climate change, silent agriculture, food trade, alternative and novel food systems, among many other topics. Just a few housekeeping announcements. Please feel free to type your questions in the Q&A and the chat box. I have activated for everyone. I'm going to do my best to address the questions that we have within the time that we have, which is 1 hour. Today, I'm very happy to present my panelists. We have Dr. Eleonora Cominelli and Professor Abdelilah Meddich. For Professor Meddich, we're going to have two amazing PhD students presenting for the SUS-Agri CC project, Ms. Chaima Ikan and Mr. Odao Redouan from Cadi Ayyad University. But for now, we're going to start our round of presentations with Dr. Cominelli. Dr. Eleonora Cominelli is a senior researcher at the National Research Council in Italy, and her research interests are focused on the characterization of seed quality in legumes and cereals, seed biofortification, regulatory roles and transport during phytic acid metabolism, evaluation of stress responses associated to drought in common bean, mineral availability from diets, among others. And she's also actively participating in projects related to agri-food production, innovative technologies, and the valorization of plant genetic resources. Today, her project title is Biofortification of Common Bean to Promote Healthy Diet and Food Security in a Context of Climatic Variation, which is part of the BioBelief project from the FOSC ERANET. Dr. Cominelli, thank you so much for accepting our invitation to present today. And I am going to just stop sharing my screen so you can start your presentation. Thanks so much.
Thank you.
I just stop here. Perfect. Thank you.
Hello, everybody, and thank you very much for this opportunity to share with you some of the results of our BioBelief project. I would like to start presenting to you the consortium of the project that is coordinated by Professor Karl Koenert from University of Pretoria in South Africa. There are another team from South Africa, the Stellenbosch University. There is our team from CNR, IBA in Milan, two other Italian teams from CREA, one in Lodi and one in Rome, and the University of Aix-Marseille. We also have two partners in kind, Embrapa in Brazil and a seed company, Blumen, in Italy. The main objectives of the BioBelief project concern the improvement of common bean nutritional quality and the improvement of common bean tolerance to drought. Of course, because 15 minutes are not so many, I will focus on the second aspect of the project, but I would like to present some topics, general topics of the project. Common bean is, of course, a legume, and you may be here about the necessity to transition towards more sustainable food alternatives, particularly based on plant-based proteins. Common bean seeds have a high nutritional value because they contain they have a high content in proteins, but also in vitamins, in minerals, and low content in lipids. But in common bean seeds, there are also some anti-nutritional, also called bioactive compounds, such as phytic acid. Phytic acid is a strong cation chelator because it consists in a ring of myoinositol with 6 phosphate groups that are obviously negatively charged. And so it is a strong cation chelator that reduces mineral bioavailability mainly of iron and zinc that are important in the diet. And for this reason, it is known as an anti-nutrient. So in the last years, many scientists in different species, different crops, isolated low phytic acid mutants. But the main function of phytic acid are storage of phosphorus on one side and also signaling on the other side. Concerning the drought tolerance, common bean yield is highly affected by drought as many other crops. Here you have the 2 examples of 2 different cultivars in which you can see the yield that is around 1.5–2 tons per hectares when the fields are irrigated. And it is highly affected by drought, with losses reaching 10 and also 100%. You can see the value and the decrease. So, in the frame of the BioBelief project, we are trying to, on one side, to improve nutritional quality using low phytic acid mutants, using genotypes with high seed iron content, and trying to combine both traits. On the other side, we are studying selected genotypes and their response to drought tolerance at vegetative, but also at the seed level in response to drought. We are trying a gene editing approach to improve drought tolerance, and the group of nutritionists that are present in the frame of the consortium are selecting the optimized bean lines and are realizing traditional recipes and characterizing them. The selected common bean genotypes into the project are 4 commercial lines, 2 from Italy and 2 from South Africa, and different experimental lines. Here you have you can see the list of all the traits of these lines. They are mainly LPA mutants because traditionally in our lab, but also at CREA GB, we isolated different LPA mutants in common bean. The activities of the project are here summarized and consist in 4 work packages. The first one in which we are performing seed multiplication, line evaluation, breeding activity, biochemical and molecular assisted selection, and genome editing of candidate genes for drought tolerance. A second work package in which we are evaluating the response to drought stress at the vegetative, but also at the seed level. There is a work package 3 for the selection of optimized beans and the characterization of cooked beans used in traditional recipes, and of course a work package related to coordination and scientific dissemination. Now I will move to the focus of my talk, in which I will explain to you how we choose and a gene to be edited in order to obtain plants that are more drought tolerant. As you probably already know, stomata are pore present on leaf surface, on leaf epidermis, through which CO2 enter and water is transpired. And plants lose 95% of their water via transpiration through stomata. So engineering of stomatal activity is a promising approach to reduce water requirement and to enhance productivity under stress conditions. Stomata result to be open in a condition of high light and well watering, while they need to be closed in response to pathogen, in response to dark, to dehydration, to some hormones like ABA, and to when CO2 concentration is high. A lot of years ago, in the lab where I worked before, we isolated a transcription factor, a protein that regulates the transcription of other genes, the first one that is involved in stomatal movement regulation. Then other transcription factors were isolated with a specific function into guard cells. We performed this work in the model plant Arabidopsis thaliana. And we found that this gene is downregulated in response to ABA, the hormone that mediates drought response of plants, and in response to desiccation. This gene is specifically expressed in GAR cells, as we found fusing its promoter to some reported genes like GFP and GAS. But more importantly, we found that ATMIB60 mutants has a decreased stomatal aperture, also in light condition. If you can see, and you can see it here, in fact, here you have wild-type plants exposed for 3 hours or for 6 hours to light, and here mutant plants in this gene. And you can see that there is a strong reduction in stomatal aperture in this mutant. At the macroscopic level, these plants resulted to be more drought tolerant, as you can see in this picture. So we started from this information and tried to identify the putative ortholog of ATMIB60 in common bean. And we found this gene that was highly similar to other MIB60 characterized also in other species, because the first paper was on Arabidopsis, but we published also a paper in which we characterized the Vitis vinifera ortholog, and another group published a paper on the tomato MIB60 gene. And in all this paper, we found that they were the true ortholog. Concerning the common bean gene, we started from an expression analysis, and we found that it is expressed in organs where stomata are present. Here you can see flowers, pods, shoot apical meristem, and the young leaves. Moreover, we prepared some stomata enriched epidermis fraction, and we found that the gene is highly expressed in this fraction compared to wall leaves, and it has a similar expression to this CAT1 gene that is the putative ortholog of an Arabidopsis stomata specific potassium channel, specifically expressed in GAR cells. Moreover, we cloned this gene under the ATMIB60 promoter and used this construct to complement the ATMIB60 mutant plants of Arabidopsis. And as you can see here the wild type stomata clearly very highly open, the ATMIB60 stomata closer than the wild type, and the complemented lines with the PVMIB60 gene are very similar to the wild type. You can see also the quantitative data in this picture and in this graph. And so we found that PVMIB60 is the true ortholog of ATMIB60 because it is able to complement the mutants from Arabidopsis. So it is a promising candidate for editing to obtain drought-tolerant common bean cultivars because as in Arabidopsis, when you switch off the gene, you have more tolerant plants. We hope that also in common bean, if we edit this gene through CRISPR-Cas approach, we hope to obtain drought-tolerant common bean. In common bean, there is a big problem about recalcitrancy to be transformed. And the only method through which people succeeded in transform common bean was the biolistic method, using shotgun and shotting DNA into embryonic axis. In fact, for this reason, we are collaborating with Professor Francisco Aragao in Embrapa that leads the only lab in the world that succeeded to transform common bean. So we prepared some constructs in which we design, in which we put single guide RNA in order to switch off through CRISPR-Cas MIB60 function under the Arabidopsis thaliana ubiquitin 6 promoter or under the same promoter from soybean. In one case, we put the single guide RNA on the single promoter. In another case, we put both in tandem under the soybean promoter. Our strategy were not very successful, unfortunately, but at the moment we have two plants that resulted to be transformed with this construct, but not edited. So we are waiting for the next generation. And at our institute, we have big problems with rooting. Of course, we reproduce exactly the protocol we learned from Embrapa. But as you know, when you change some conditions in your experiment, sometimes something does not go in the hoped way. These are the results for the moment on this transcription factor. I would like to acknowledge all the entire BioBelief consortium, particularly Professor Karl Pruner to coordinate the project, and Vera Martinez Barrada and Patricio Arce Johnson, with whom we collaborated for this specific part of the project concerning PVMIB60 from Pontificia Universidad Católica de Chile. Thank you for your attention.
Thank you, Dr. Cominelli, for this fantastic presentation. Like, I'm a plant biotechnologist, and I was like, oh yes, this is my thing. So I just want to remind people you have the space to ask questions in the Q&A box, in the chat box. But in the meantime, I have some questions. So are you testing the system in like different varieties, and like how do they work? Like if you do in tissue culture, like is there like differences on how they regenerate? Like I know this is a pain, I know.
Yes. It is a very good question. The answer is yes, because we are using the same variety that is used by Professor Francisco Aragao, but the group from Pontificia Universidad Católica de Chile is using another variety. So we are, for brevity, I didn't tell you many details, but we are working in parallel using two different varieties and using different tricks in the protocol. For example, now we added active carbon in order to eliminate catabolites, as suggested by Professor Patricio Arce Johnson. So we are trying to move on both sides, but it is very tricky.
Yeah, it's always with tissue culture.
Yes.
And so when you the ploidy of these beans, are they diploid or are they polyploid? I don't know.
No, they are diploid.
Ah, okay.
Yes, they are.
And then just like that's if it's diploid, you should, because when it's polyploid, it's kind of when you do the gene editing, you just like have many copies of the gene, so that's kind of complicates more the stuff.
Yes,
But
of
yeah,
course. But the main problem is routing.
it's routing. Okay.
Three step.
Mm. So, and do you know what, like, what's the at the moment, like, what's the landscape on gene edited like crops at the moment, like in Europe? What's the stance? Do you know, like, what's happening? Like, we are allowed to experiment with them, but not to release them, is it?
Yes, it is quite complicated. There are some opening compared to the transgenes because they are not considered as transgenic plants because you can completely remove the exogenous gene you introduce. Then it depends also on the single countries. For example, in Italy, we are more restrictive than in other countries.
Yeah.
And the legislation from Europe has to be reached, and not in all the countries there is the same response. But we hope that we are moving in the right direction to approve this kind of modification because they are very useful and they are very promising. We are trying also to use the appropriate names to call them. For example, in Italy, we call them TEA. Techniques of assisted evolution. So in order to
make it more appealing.
Yes, make them more appealing.
And in your experience, so with traditional breeding compared to genome editing, what's the time difference more or less? Like I in bean, I have not worked with beans, so what is the time difference of you to be able to get like a modified like a breed variety with the qualities that you want compared to genome editing? What would be the time difference?
Yes, of course, in the case of common bean, I can be very optimistic because we have these problems in regenerating plants. But if you use a crop in which the transformation is a routinary approach, like for example rice, of course, to obtain some results through breeding, you need a lot of generation in order to fix a particular trait. Of course, if the editing approach and the transformation approach is quite easy, the time are very, very reduced. I'm doing the example of rice, but I do not know exactly the difference in terms of times. But I have some colleagues at university that now make CRISPR-Cas use CRISPR-Cas approach in a very routinary way, and in some months they obtain the modified rice
Yeah, this is pretty quick now. So is there a part of the project where you guys are trying to um encourage people to consume more beans to make it like a more integral part of the diet?
Yes, it is, because we have the two groups of nutritionists, the one of Crea Ayyad in Rome and in Aix-Marseille University, that are doing also an activity of dissemination in order to promote legume consumption in general, in particular common bean, through different activities, and they are also trying to understand the best of the lines we are using, or the best genotypes, and there is an activity of promotion of these use. Unfortunately, in our project, another partner would have to be Kenya, Sia from Kenya, but there were a lot of administrative and bureaucratic problems, because there was a specific part in the project attributed to Kenya for this promotion of common bean consumption, but unfortunately this partner maybe in this last month of the project can probably join, but there was very important problems about this partner.
Wow, interesting. We have a quick question here from the audience. It says,
Yes.
does the bean easily allow for gene transcription? Do you encounter some differences? I imagine like the transcription of the new genes that you introduce, as in if when you do qPCR, you can like see that the genes are expressed. I imagine that's the question.
Yes. No, we do not have any problems to check transcription of genes in common bean.
Okay, so you put the gene and they express, and that's fine.
Yes.
Okay. Okay, that's perfect, Dr. Cominelli. I think that's all the time we have, because we're gonna move to our next panel. Thank you so much for
Thank
accepting our
you.
invitation. And like, I hope Siad is able to join the project soon, and then to see like more results for this project. Thanks so much.
Thank you very much.
Thank you.
I
And
stop my sharing. Okay.
yes, perfect. Thanks so much. And now we are going to have Professor Abdelilah Meddich. Professor Meddich is a full professor and researcher at the Cadi Ayyad University in Morocco. His research interests focus on the sustainable and climate-smart agricultural practices using biostimulants and innovative biofertilizers. His work explores the role of arbuscular mycorrhizal fungi in enhancing plant growth, physiology, and biochemistry under water stress and heavy metal contamination in different environments. Additionally, his research includes the remediation of heavy metals using plant microorganism interactions, contributing to innovation in sustainable agricultural practices. And today, his project is entitled Innovative Biofertilizers Boosting Yield of Cereals and Horticultural Crops Under Global Climate Change towards sustainability of agricultural systems against climate change in arid zones, which is part of the SUS Agri-CC project from the FOSC ERANET. And today, this project is going to be presented for his two fantastic PhD students, Chaima Ikan and Redouan Odau. Thank you everyone for accepting my invitation to join this webinar series and to present your projects. And I'm just gonna stop sharing my screen and you can start, guys, the presentation. Thanks so much.
Okay, good morning everyone. I'm pleased to be with you today. Please allow me to present to you the state of progress of our research study entitled Innovative Biofertilizers Boosting Yield of Cereals and Horticultural Crops and their global climate change towards sustainability of agriculture system against the climate change in arid zones, presented by Ayyad Dordowan and me, Shayma Ikan, and supervised by Professor Meddich Abdelilah. To better follow this presentation, the outline was structured as follows. We will start with a brief introduction, and then which will be followed by main objectives and material and method section. And then we will move to the results and finally conclusion and perspectives. Today, more than ever, we are facing an exponential increase in the world population, which will reach by 2015 9 billion. This means that the need for food has increased, and which pushed farmers to use excessive chemical fertilizers. The use of this dangerous input have also aggravated the situation. And parallel to the increase in the population, the intensification of climate change is leading to the scarcity of water, which is aggravating drought. Globally, Arab region is the most water stressed in the world, and in Morocco, water stress is between 25 and 70%. And also, Morocco is one of the around 30 countries condemned to face extreme water scarcity by 2014. For this reason, it is necessary to think about new sustainable approaches, such as arbuscular mycorrhizal fungi, compost, plant growth promoting rhizobacteria, which they have for role, they play as an alternative to chemical fertilizers. They can improve soil aggregation, improve nutrient and plant growth, and also restore degraded soil, ultimately to ensure food security. Therefore, the main objective of this research study are the evaluation of the efficacy of combined natural biofertilizers on the tolerance of cereals and horticultural crops and their drought stress. Secondly, determination of the agrophysiological, nutritional, and biochemical parameters of crops, and then understanding the molecular mechanisms involved in crop tolerance to abiotic stresses via omics analysis, in particular metabolomics, proteomics, and hormonomics. And finally, the determination whether biofertilizers applied alone or in combination can compete with chemical fertilizers to improve the growth of different crops under water-limited conditions. In order to achieve the mentioned objectives, we have adopted the following methodology. A trial experiment was conducted at Loudaya site near to Marrakech city with two water regime, 100% crop evapotranspiration for the well-watered regime with a flow rate of 8 liters per hour, and 30% crop evapotranspiration for the drought-stressed regime with a flow rate of 4 liters per hour. Different treatment was applied, positive control with NPK, negative control without any biofertilizers, and then mycorrhiza, compost, and PGPRs applied alone or in dual or triple combinations. For the drip system irrigation, we have used the famous equation, which is crop evapotranspiration equal to crop coefficient multiple of reference evapotranspiration. For the vegetal material, it constitutes of tomato and lettuce seeds for the horticultural crops, and maize, quinoa, and wheat for cereals. For the biological material, we have used PGPR consortium based on two strains, IMF consortium, and also local compost. Here we have some illustration from the plot preparation to the application of biofertilizer and then transplantation of different crops. Several parameters were measured, such as mycorrhizal parameters, like frequency and intensity of mycorrhization, biochemical parameters like protein, sugar, and proline content, photosynthetic pigments, malondialdehyde, antioxidant enzymes activities, and fruit quality, also growth parameter like number of leaves formed, root length, shoot height, total fresh matter, total dry matter, and yield. And we have also noted the harvest date of different crops for the first trial and also for the second trial. Here we have some other illustration about PGPR inoculation, about some phenological stages of different crops, and also some measurement of soil moisture and chlorophyll fluorescence can change, and also the visit of the project partner from Mexico, from Morocco, and from Japan. Now we will move to the next, to the results section, which will be presented by my colleague Radwan Ohito.
So hello everyone. Let's start now with the results of the first trial in 2023, starting with the results of maize. So as you can see in this table, we noted a significant effect of biostimulant on yield and its characteristics under drought stress. So the application of PGPR alone or in combination with IMF improved cob length and cob fresh weight. We noted also an improvement of yield by the application of triple combination, which means the association of compost plus IMF plus PGPR. Physiologically, drought stress affect leaf area and normalize differences vegetation index, tomato conductance, and the chlorophyll fluorescence. However, we a noticeable effect on leaf area and in DVI was observed in plant treated by PGPR plus IMF plus compost under drought stress. Somatal conductance also and chlorophyll fluorescence were improved in all biostimulated plants compared to the control plant. The same biostimulant positively affected the biomass and mycorrhization rate of lettuce plant. For example, plant treated by compost had a higher biomass than control plant. Rate of mycorrhization was enhanced in plant root inoculated with IMF plus PGPR in the presence of compost. Drought stress also affect physiology by decreasing water content and stomatal conductance, and biochemistry by increasing hydrogen peroxide or H2O2 and the proline content. So in plant treated by C had a higher water content than control plant, as you can see in this figure, and stomatal conductance was significantly improved in plant inoculated with the IMF alone compared with control plants. H2O2 or hydrogen peroxide was reduced significantly in mycorrhizal plant by 38%, while the NPK increased proline content. So, introducing biostimulant in the wheat field improved the yield. For example, in this figure, we noted that short dry weight and green yield were improved significantly by the application of compost plus IMF under drought stress. Also, raw dry weight and straw yield were enhanced by when plants are treated by IMF plus PGPR and PGPR alone, respectively. So we note that also the drought stress modified the biochemistry of wheat leaves. We can observe that drought by the application by using biostimulant affect protein and box, and they can modify them by improving significantly this compound by the application of compost plus PGPR under drought stress compared to the control. But plants untreated had a higher values of total soluble sugars and proline than treated plants. For tomato also, in the same conditions, we observed that drought stress modified the physiology and also the biochemistry of tomato. In these figures, we present the yield and the number of fruit, and we note that under 15 ATC or person ATC plants inoculated with IMF have a high yield around 250 kilograms and the fruit number compared to the control. Also concerning the quality of tomato fruit, we note that drought stress generally increase polyphenols and the flavonoids and lycopenes, especially when tomato plants are cultivated in amended plot in the presence of AMF compared to the control. In contrast, drought stress decrease also carotenoids content by the application of compost and IMF. So here we put some illustrations for the second trial in 2024. For example, here we can see the corn seeds emergence, measuring chlorophyll fluorescence of lettuce and tillering wheat stage, and also other plant, for example, PGPR inoculation. This slide set up the calendar of crops in this season. We observed that wheat and lettuce were harvested in May 2024, and we are now in biochemical analysis stage. For tomato and quinoa and crops, they are now in progress, and we do just a monitoring of physiological parameters and growth, notably somatal conductance, NDVI, and chlorophyll fluorescence, and all these crops will be harvested in the beginning of July. So as a conclusion, the application of IMF alone or combined with PGPR in amended plot increased growth and yield and improved physiological and biochemical parameters of cereals and horticultural crops compared to the control under drought stress. This tolerance due to an increment of proline and total soluble sugars, and by reducing also stress markers. So we can say that the use of biostimulant alone or in combination is a practical alternative to the chemical fertilizers application for improving cereals and horticultural crops tolerance to drought stress. So this biostimulant can constitute a promising approach for improving the tolerance of crops under water deficit condition. And thank you for your attention.
Thank you so much, Chaima and Redouan. I just want to remind people that they have opened the Q&A section and the chat section to put their questions. So while people are like typing away their questions, I'm not like, this is not my field. So when you talk about the concept of mycorrhization rate, how do you actually measure that? Do you have to remove the plant and measure how much mycorrhiza has been formed, or?
No, after the harvest, we separate the part, the aerial part, and the root part, and then after that we wash the roots, and we apply a standard protocols of Heyman, and after that we move to the coloration of roots. And we start to do some observation in microscope, and then we can calculate this parameter.
Okay, so it's some kind of counting under the microscope.
Yes, of course.
Okay, I see. So you mentioned that maybe one of your treatments, like for example, AMF alone, or let's talk about that one just. How do you think it would be easy to scale up a method, like a system like this AMF massively, if you want to use it like in many fields, like commercially? What do you guys think?
I think we are now in the process of testing our biostimulant. and after that we can pass or move to the practical application by, for example, IMF. We can multiply them. We use, for example, the maize crops for multiplication of IMF, and then we can use a lot of quantities for the application of, or we can also commercialize them.
Okay.
But sorry, I can just add one information
Mm.
about the feasibility economic. I think for bacteria PGPR, there's no problem. There is the equipment that we can multiply a large scale PGPR. The same for compost. We are calculating some economical possibility to have it in large scale. But about mycorrhiza, it's not easy in our case. We are in discussion with our partner. It's not easy. Yes.
Okay, so you have a partner who is trying to produce
Yeah.
it in large scale and is a bit difficult for the
Yes.
mycorrhiza. Ah,
Yes.
interesting. Interesting. So for, let's say, talking with like our PhD students, so guys, what do you think we will be like the next steps in your research? Like what would you do like to go like ahead in your research? Because I think you are almost completing your PhD. So what do you think you will go next in this kind of research?
I think, sorry, Radwan or Shaima, for the next step, we will, in the second campaign, we will confirm the performance of our biostimulant, and we will focus in one, I think, on three-party combination between IMF, PGPR, and compost. We are in discussion with other partners, Japan, France, and other partners in University Mohammed VI Polytechnic, for having the possibility to pass and to achieve some step from. We are now, I think, in 4 or 5 TRL. So we hope to go to 6 and 7 TRL. But we now we have some formulation that give good work, not only in laboratory, not only in one feed, but in two feed, in two compound, etc. So we are in this position to pass our research from development to deployment to investment. It's not easy, but we do our best for this way.
Yeah. And how do you see the implementation of these kind of biofertilizer systems in farmers? Like would they have there been talks with farmers around there to see if it's not like, because sometimes these kind of things, it might bring more work to them or not? Like what is their take? What do they think about these kind of approaches with the biofertilizers?
Yes, we continue all time to involve the agriculture and the farming. And in this project, SwissAgriSeed, we have, I think, just one seed and two compound to be explored. But we have, I think, more than 10 projects in parallel that we continue to work together with agriculture. We believe to the seeing and believing. So we continue to, I think, in the next semester of the 2025, we will have a big workshop that we invited all the partners, and especially the agriculture population.
Oh yeah, that's great. So you're going to kind of show the results to the farmers, and they'll be like, yeah, probably happy to see that they can use something else for their soil and to grow their plants. We have a question here from the audience. It says, thank you for your presentation and progress. I wonder if there is experience on recommendations of mycorrhiza for crop production.
Yes, we have some difficulty about the technique of multiplication at large scale about mycorrhiza, but some advice that we give quickly. The performance of mycorrhiza is best expressed in the case of soil poor in organic matter and nutrient. So we can't explore the efficiency or performance of arbuscular mycorrhiza fungi in the soil rich in nutrient, mineral, or organic matter. This is the best way. The second advice that we can give, we have some difficulty when we combine PGPR and compost. We should be careful about some doses of compost. If we increase the dose of compost, we lose the performance about our inoculum. And there is a lot of mechanism possible that we found in our work. These two scenarios of increasing crop tolerance to stress by mycorrhiza and additive organic or inorganic that we can share with our PhD student or other scientific community. But the best advice, we can't use and explore mycorrhiza in the soil rich. They work especially in the hard condition.
Interesting, yeah. We have another question here. It says, despite research clearly showing the potential of this technology, what are the major limitations that are stifting its widespread adoption?
I think there are a lot of limitation. First, sometimes we work now in 5 plot, different 5 field. So we have some difficulty. Sometimes we have a good tripartite combination between mycorrhiza, compost, and PGPR. When we change the field, we have another result. We found that the bipartite combination, for example, mycorrhiza and compost, will be the best in another field. Why? Because there is some change in weather, in climate, in temperature, in precipitation, in texture of soil, in the quality physical-chemical of soil. There's a lot of parameters that can alterate or modify the performance of our technology. And in large scale, there is some difficulty sometimes. Until now, we have to move to the metagenomic and other omics to detect where is the efficiency is related to compost sometimes, or to PGPR, or to mycorrhiza. So we're working about defining these or to give the answer of this question. We don't know where is the best entity that works in these 3 combination. There is some specific mode action, and there is some synergetic action.
Yeah.
And other things, I think in terms of feasibility economic, we have other to join other partner management and other specialty in economics in industry to have the possibility to go from tier 4, 5 to 7 or 8. There's a lot of limitation.
Yeah, and it's like a lot of variables, like in these kind of experiments.
Yes, of course. I have one limitation. Sometimes we start to isolate the microorganisms in the soil. For example, the bacteria.
Yeah.
We start to do some tests. For example, solubilization of phosphate and potassium, and the secretion of some phytohormones like auxins. And after that, we can find that this bacteria has an aspect, and pathogen aspect.
Oh.
So we have to pass toward the molecular analysis before apply them in the field or next to the plant.
Yes,
Yeah.
I confirm. In laboratory, we have some best performance strains of bacteria, but after analysis molecular, we find them that they include some strains which have that very pathogenic. So we don't use it in field. So in field, we use just bacillus, benefic bacteria. Yes.
And has it happened to you that you maybe are trying to identify an organism and then you have it in soil, but when you try to kind of culture it in media, it doesn't grow?
You can repeat the question?
That when you identify, like you have in soil, like you have identified, like, I don't know, a
Yes.
bacteria that could be a potential. And then when you go back to the lab and you try to reproduce and grow it and have it in plates, it doesn't grow. Because I know this happened before when they're in the wild, they grow, but when you try to bring them to the lab and increase their production, they don't grow.
No, so sometimes we have to be careful before apply them in the soil. We have
Okay.
to be careful when you apply them in the labo, and also to transfer them in the soil next to the root,
Okay.
because they can pass also to the home
Okay.
or to people.
And
Yeah.
other information about this bacteria which present a pathogenic aspect, we maintaining them in laboratory, and we are in collaboration with Japan partner. So they have a lot of best-way about solubilization, phosphate, potash, Ayyad. So we can explore in them for about other protocol. Yes, we don't use it in the feed, but we can explore them in other very interesting protocol. In other stress in laboratory.
Okay, okay, I see. So there is a way to kind of do it also in a laboratory setting. That's very interesting. We have a question here from a member of the audience. It says, have you worked with multiple organisms, single PGPR formulation, in a single PGPR formulation? And if so, how their performance compared to the use of single organisms?
Shame you can give an answer. If not, I can give it. In our work, we prefer to work with consortium when compared with single application. In mycorrhiza and same in the PGPRY, because when we work with consortium, more than 1 or 2, 3 species, before that we tested their confrontation. There is no inhibition between the strains. So with consortium, with a complex mycorrhiza or complex bacteria, we have more chance to have the best formulation when compared with single strains. It's true in PGPR and also in mycorrhiza.
Okay, perfect. Thank you so much, Redouane, Chaima, Professor Meddich, for your fantastic presentation and for answering all my questions and the questions from the audience. This has been an amazing set of 2 presentations, and this is the last one for the summer, and we're gonna have more starting on September, and I'm just gonna send the invitation to everyone who also attended this one. So that's it for me. It's 10 a.m. So I'm just gonna release everyone to go and finish your day. And thank you so much for joining everyone, Dr. Cominelli, Dr. Meddich, Redouane, and Chaima. Thanks so much. And it was nice to meet you all.
The same.
Thank you. Bye-bye.
Bye-bye. Bye-bye.
Bye.
Thank you. Thank you.